Hello, and thank you for joining us for the Advancing Science Spatial Genomics Data Analysis Scientific Track. I'm Jason Reeves, Senior Scientist in the Translational Sciences Group at NanoString Technology, hosting our program today. Joining me are several leaders in the field driving forward spatial omics research, including Chris Mason from Weill Cornell Medical Center, where he's an associate professor and co-director of the World Quant Initiative for Quantitative Prediction. Dr. Christina Curtis, associate professor and co-director of the Molecular Tumor Board at Stanford University Cancer Institute. Dr. Omer Bayraktar, group leader for the Cellular Genetics group at the Sanger Institute, and he will be joined during our discussion by his graduate student, Alexander Aivazidis. And finally, we'll be joined by Dr. Ioannis Vlachos, who is the co-director of the Bioinformatics program at the Cancer Research Institute for Beth Deaconess Medical Center. During this track, each presenter will present their experiences with genomics DSP and their institution's perspectives and approach to spatial biology and data analysis. This will be followed by a roundtable discussion with all speakers to gain greater insight on data standards, as well as the infrastructure and resources required to execute on a new wave of technological development within the growing omics ecosystem. As a quick housekeeping note, if any questions arise during the presentation, we encourage you to submit them to the Q&A box below. We will respond back with your questions via email. Please join me in welcoming all of our presenters, and I would like to hand it over first to our first speaker, Chris Mason.
Thank you so much for the introduction. It's really a pleasure to be here with a really extraordinary group of investigators and clinicians here. So I will jump right into some of the work we've been doing for shotgun transcriptome and spatial profiling of COVID-19 and what we see what the virus is doing in the body and around the body and how it manifests, basically. So basically, I'll start with a quick preview of some work that we've put in a preprint in a paper that's in review of taking the first 735 patients that came through the doors at Weill Cornell Medicine and New York Presbyterian and did RT-PCR, LAMP, as well as total RNA-seq to look at the differences between the COVID positive and negative patients. And this will set the stage for what we wanted to look for as a deeper dive in some of the genomics data that I'll show you here today. But basically, we wanted a variety of molecular methods to do a dive on what's happening when we see the infection. Can we pull out viral sequences and perform phylogenetic and genetic analysis of these variants and see where they've come from? And indeed, we could actually see, basically taking all these fragments of RNA, align them to the reference genome compared to the Wuhan strain, and we can see here that we can reconstruct some of the phylogeny and evolutionary history of the virus, and so a lot of the different variants are showing up in some of the New York City samples from the bulk RNA-seq data alone. This also let us look at the presence of co-infection. So if you look here, we can actually see whether there was low, medium, or high levels of SARS-CoV-2, and if there wasn't on the right, you can see this sort of pink section where we could pick up other respiratory and RNA viruses as well. And on the left, you can see in some cases pneumonia was also present. So we can see from the total RNA-seq data, we can discern the infection broadly. We could also look at differentially expressed genes, or DEGs, and see upregulated in orange and downregulated here in purple, from this bulk RNA-seq data, and profile interferon-stimulated genes and generally inflammation networks that if you look from a pathway perspective, would group into things that are familiar, such as reactome interferon signaling, antiviral response. But some that were surprising, such as olfactory signaling pathway here in the lower right, or hemoglobin and iron regulation, indicating some of the really cardiac and hematological disruptions from this virus, which were really surprising. We also built out tools to look at cytokine interferon signaling and make these heat maps for any select set of genes. And actually, one of the things I want to quick highlight today is that we also have an online interface where you can take some of these same data and make your own heat maps and explore our data. It's a website we put up at just covidgenes.weill.cornell.edu, which is for anyone to explore the data. We wanted to make this as open resource as possible for anyone to play with these data, as well as some of the isoform characterizations. And so we use some of these data, and actually I've already published one paper just looking, for example, at complement factor pathways, which you could see some of the same kind of disruptions from these SARS-CoV-2 infections. And so what's interesting is, some of these has been, this has been published in a variety of contexts and preprints, some of our own, in the past 4 or 5 months. But what's really missing from this is, we can see a lot of the disruption in the body. We know that there are really challenges that the body has to confront during infection. But what's been missing, of course, is a more granular and even spatial characterization of what happened during the infection. So we actually have been collaborating with NanoString we just got our genomics installed right before COVID hit New York City. So it was a good timing. Also collaborated with them, have some samples run out of West. And what's, I think as most of you will cover in this webinar, is that this gives you an ability to use really high plexi mixtures of reagents that have a UV photocleavable linker with the DNA oligonucleotide, as well as RNA reagents with an oligo-labeled probe that let you get the spatial information using the genomic system to look at, say, FFP slides, which is actually very important for the COVID-19 autopsy samples, which I'll show you today, because we needed to put them in FFP because they were still infectious if we didn't actually put them in FFP or some sort of shield or trizol. So other systems where you can't use FFP-treated samples basically were intractable in the early days unless you had a BSL-3 lab. So what we've been using is the genomic system where actually then illuminate and UV cleave some of those regions of interest, or ROIs, or areas of interest, or AOIs. And then we can actually, this is a slide just based on the genomics. You can see we can then look and see the expression levels of what's present in different regions across these tissues. So what we did is, as I alluded to, use the stain slides with the oligoconjugate antibodies, look for ROIs, and then also wanted to do spatial profiling as well as cleaving, putting them into plates, and then also sequencing them on NextSeq, and then characterizing them essentially with accounts per gene across the different regions. And so, the workflow basically is DSP library prep is pretty standard on a NextSeq 550, and then sequencing and counts of analytes per gene, which I'll show you some of this data today. And this was a very collaborative effort because we did this between two different sites. So we did some Zoom calls with NanoString and went over some of the logistics. And we wanted to look at regions of interest from a large airway, vascular zone, alveoli, and also macrophage infiltrated tissues from autopsy patients, from the first 35 patients that got autopsies done at New York Presbyterian, to get a sense of the tropism of the virus. For example, where is it going in the body? And then what is the virus doing at a spatial level during the infection, to better learn about the virus, and also hopefully to figure out ways to then combat it. So what we did is, conferred and then selected these ROIs in collaboration between actually Rob Schwartz here at Cornell, and also my lab, also Alan, who's at Cornell, and then NanoString's group out West. And basically picked 285 areas of interest, basically filtered for any samples that had low reads or problematic uniqueness criteria, and then basically had about 282 AOIs that were left. And if they had fewer than 250 nuclei, those were removed, or those that had fewer than 50 genes identified. So kind of some basic quality control of these sections we got from the alveoli, large airways, and some other parts of the sort of samples we got from the autopsy. And you can see down here on the left, the number of genes detected per AOI was anywhere from 500 to 1,000, or in some cases 1,500. And the number of, what we see for the scan for the different samples, a lot of them we're getting from the alveoli sections you can see here on the right. But overall, seeing that range of detected number of genes per AOI. And we could actually pick up here is of the, about over 1,800 genes in total, we got about 18 or 11 from the CTA assay, which is a comprehensive cancer transcriptome atlas assay. And also we did look at some viral genes to spiking to look for COVID. And again, also did some QC filtering here on the data as well. You can see just for basic QC. What's interesting is when we looked at the samples that had COVID versus flu versus normal and acute respiratory distress syndrome, or ARDS, we can see that the normal and flu samples were clustering pretty well in the alveoli, whereas there was some more variegation and heterogeneity of COVID and the ARDSs based on these samples. And then when we looked to see if we could recapitulate some of the same pathways that we observed in bulk RNA-seq data, we could indeed see some of the cytokine signaling that we saw in the immune system, as well as interleukin 4 and 13, basically alpha and gamma signaling from interleukin response to signaling pathways, which show up actually in some of these samples for the gene set enrichment. The other interesting thing, and so this is something you can get from bulk RNA-seq data, so it was good to see some of these gene set enrichment analyses, or GSAs, again, recapitulating these data. What was really exciting, I think, was to be able to tease out the abundances of different samples, whether they're COVID samples or flu or normal or ARDS, and see things like this huge burst of macrophages in some of the COVID patients, or, for example, other variations of disruptions of what you'd see for neutrophils would be, compared to, say, normal samples, which are actually the normal lungs that we got here were from lung transplant patients, where, as you can imagine, when you basically do a organ transplant between people of different sizes, some people's lungs are bigger than others, so you have to shave them off like at a deli, and these are what we use for normal samples. So it's an interesting thing about normal, but clearly some of the COVID samples are very, very different. Then to validate these, we used RNA-scope as a method to characterize this with a different hybridization method. And this basically was looking at the COVID flu in normal samples to look for the presence of ACE2, basically receptors for ACE2, TMPRSS2, and also the presence of the virus itself. And so from these samples, again, we looked at COVID patients, flu, and then one normal, some preliminary data to see what was detectable. The other targets were detectable at very low levels or not detected. And to lay this out for you visually, we can see, for example, ACE2 expression and also, in some cases, presence of the virus at low levels from the RNA-scope versus genomics data. And we can see at high, TMPRSS2 was, in some cases, very visible, but not as much present in the alveoli, for example. So we could see some heterogeneity in the expression as expected at a spatial level for these different markers. And you can also see, in some cases, the virus present below the levels. This is one case that actually knew based on RT-PCR didn't have much virus. And again, we didn't really see it here as well, but we could see plenty of TMPRSS2 in both the large airway and the alveoli. As another example of seeing variegation of TMPRSS2 in the large airway and alveoli, especially here at the sort of rim of some of the alveoli. So again, underscoring some of the heterogeneity that can be, and we do see in some of the COVID patients, as well as the way they deconvolute the cellular sort of infiltration and sort of damage that's occurring in the lungs. So in a quick summary, this is, we could see differential clustering of expression where the COVID and the ARDS group seem to see more heterogeneity. We could see TMPRSS2 definitely highly expressed in larger airways, but variable among COVID samples, and also the virus variable within the lungs, especially later in infection. We're less likely to see the virus present in the lungs. And also we could see single-cell deconvolution methods that could see, for example, infiltrating macrophages or neutrophils that would be much more pronounced in the COVID patients. And I want to thank NanoString as well as thank everyone in the lab who made this possible, and a great group and really inspiring team who we often like to go throw axes and drink beer, at least back in the pre-COVID days when we could go do this, and we look forward to doing this again sometime soon. And thanks, of course, to the funding from the NIH, also collaborations from World Quant and Citadel, as well as, of course, NanoString. And thanks, everyone, in the lab specifically. And thank you so much for your time.
Great. So I'm really pleased to share some of the recent work that we've done using the NanoString Genomics platform to understand how the tumor immune microenvironment changes through therapy, particularly in the context of HER2-positive breast cancer. So we've had a long-standing interest in understanding how tumors evolve through therapy. And in this particular context, we felt that a study of the changes in the tumor microenvironment during preoperative or neoadjuvant therapy would be particularly informative. And we sought to address this question in HER2-positive disease, where routinely speaking, patients are treated with standard of care neoadjuvant therapy. In this case, we turned to a clinical trial cohort where we were able to obtain pre-treatment baseline tissue biopsies prior to the administration of any therapy. Subsequently, these patients underwent one cycle of HER2-targeted therapy with either trastuzumab and or lapatinib, and they were then, at that point, obtained a second on-treatment run-in biopsy. And finally, these patients went on to obtain 6 cycles of HER2-targeted therapy prior to surgery to determine whether they had achieved a pathologic complete response or not. And so the neoadjuvant setting really provides a rich opportunity to obtain high quality tissue. In this case, we focused on profiling formalin-fixed paraffin-embedded material from some 56 patients split into a discovery and validation cohort from the TRIO-B07 clinical trial. And using the NanoString Genomics DSP platform, we used a phenotypic enrichment strategy where we profiled a minimum of 4 different regions of interest across each time point for each patient. And using pan-cytokeratin based enrichment strategy, we were able to specifically enrich for tumor-enriched regions. And this has tremendous value as we think about profiling relatively infiltrated breast tumor cells, but also allows us to go after the immune population that's particularly infiltrated the tumor. And separately, we can obtain the inverted mask area, focusing in on the tumor microenvironment region by taking the inversion of this. And so we profiled a total of 43 analytes per section. And to give you a sense for these data, what I'm showing here is individual patients labeled along the x-axis, where we can see whether they achieved a pathologic complete response in red, or if they didn't. We've also indicated their ER and PAM50 receptor status. And then shown along the y-axis, each dot represents a region of interest, where we're showing the protein expression levels for HER2 in blue and CD45 in red. And we can, of course, look at these ROIs and the extent of heterogeneity across each sample. So just looking at the baseline time point, we actually don't see really dramatic differences between these markers. And that was, in fact, true of virtually all markers we examined. In contrast, when we compare the pre-treatment time point to the on-treatment time point, looking at all tumor markers, we see a pretty dramatic increase in the extent of heterogeneity, with HER2, the target of these therapeutic agents, being the most dramatic, but numerous other tumor markers also increased. And somewhat surprisingly, we also see a dramatic increase in the heterogeneity of immune markers from the pre-treatment to on-treatment time point. So when we look at what's taking place overall in these individual tumors, what we can see is, in fact, by looking at a volcano plot where we're showing the change in expression in the pan-cytokeratin-enriched regions of the run-in versus baseline time point, and then plot the negative log 10 FDR-adjusted p-value, what we can see is that a variety of tumor markers, in particular HER2, but all of its downstream targets, are really dramatically decreased on treatment. And in fact, we see an enrichment and upregulation in a variety of immune markers. And just looking at a correlation plot, we see that, in fact, many of the tumor markers are very tightly correlated and anti-correlated with the immune markers. So motivated by the fact that we saw pretty dramatic differences, we then sought to stratify the discovery cohort by responders, those that achieved a path-CR and those that did not. And what you can appreciate from this plot is that really the vast majority of the signal is being driven by the responders. Again, we see that dramatic reduction in HER2 signaling by the on-treatment time point, and this is just after one cycle of targeted therapy, and a really dramatic increase in immune markers. And in contrast, this is simply really not very evident in the non-responders. And so we were also interested in to what extent this was reflected in bulk gene expression data. And what you can see here using matched patient samples is, in fact, that the signal is really dramatically attenuated. There is some signal in the PCR cases, but it is to a much lower level. And presumably this reflects the attenuation due to the admixed population. But also keep in mind that we had performed a pan-CK enrichment to really focus in on that population using DSP, and that's one of the great values of this approach. So I also want to just turn to the pan-cytokeratin adjacent regions where we can really profile the tumor immune microenvironment that surrounds the tumor, and to point out that using this phenotypic enrichment strategy, we can actually now ask which immune markers are enriched in the infiltrated tumor, so higher in the pan-cytokeratin enriched regions, versus those that are instead perhaps higher in the surrounding tumor excluded regions. And what we'll see here is that there's a number of immune suppressive markers, PD-L1, IDO1, that are increased in the pan-cytokeratin enriched regions. And in fact, we see evidence for an enrichment of T-cell markers that seem to be excluded from the tumor cell. So we can actually ask how these patterns change, not just at the pre-treatment time point, but on treatment and by the time of surgery. And what we can appreciate from this is actually that there seems to be an influx of T-cells, and this trend towards exclusion decreases over time. And so collectively, by using DSP, we're really able to address somewhat unprecedented questions in the context of the temporal changes in the tumor immune microenvironment through therapy. And so with that, I'd like to thank the many people who have contributed to this work, in particular Catherine McNamara, who read it, and it was really a very rich collaboration with our colleagues at NanoString. And so with that, I'd like to hand the baton over to Omer Bayraktar from the Sanger Institute.
Thank you, Christina, for the introduction, and it's a pleasure to join you all today. I'm a group leader at the Sanger Institute, and today I'll tell you about how we use the whole transcriptome atlasing version of NanoString that allows you to measure the expression of 18,000 genes on the developing human brain to understand the human brain tissue architecture. So in general, our lab is interested in studying the structure, function, and diseases of the human nervous system. And one of our focus points is the different cell types, their distributions, and their fates in various neurodevelopmental and neurodegenerative disorders. When we started this collaboration with the nanostring, we were actually quite excited to take the WTA assay that effectively allows you to do transcriptome-wide measurements and actually apply it to the human fetal brain to be able to distinguish different cell types. So there are many things that I can tell you about the structure of the human fetal brain, but one of the major reasons that we used it for our study is that it shows a really striking spatial arrangement of different cell types. The brain structure that we focus on is the human cerebral cortex, or also called the neocortex. And when you look at it in the developing fetal brain, you basically see that it's split into two different areas. First, as you can see on the diagram in the middle, you have different kinds of neural stem cells and progenitors shown in green, red, and blue in germinal centers. And second, you have differentiating neurons and glial cells that are found in the opposite cortical plate. And the structural and cellular development of the organ, despite its immense cellular complexity, is actually based on simple principles. You have neural stem cells and progenitors that proliferate in the germinal centers. They give rise to neurons or differentiating glial cells. These cells then migrate out into the cortical plate. So if you were to sort of spatially profile this organ across the spatial axes, you end up revealing this basically spatial axes of cellular migration and differentiation. In our experiments, we initially used WTA profiling to examine bulk regions of interest that correspond to different compartments of the germinal centers and the cortical plate. This was a great piece of work done by a postdoc in my lab, Kenny Roberts, with the great support of Robin Froth from NanoString. When we did this, we actually saw really great QC results in these regions of interest that I highlighted in the middle in panel D. From as little as 5 cells, we could distinguish the expression of at least 4,000 genes. On an average, many of these ROIs that contain a few thousand nuclei expressed 8 to 10,000 genes over limit of detection. But today, what I'd like to tell you is very little biology and mostly focusing on some of the tools, computational tools, that we developed to analyze this WTA data. So the first tool is a probabilistic generative model that we call in situ cell tools, or ISU for short, that estimates and corrects the background binding in the WTA assay. So as most of you are probably aware, WTA assay and the DSP assay uses probes or antibodies to quantify the expression of their targets. And when you rely on probes or antibodies, you're going to be dealing with a certain amount of background. To be able to identify this, Alexander Ivazidis, who joins me for the panel today, actually developed this model that tries to distinguish the biological signal that you see in the nanostring, the real signal, from the technical noise, which comes from both non-specific probe binding and the sampling noise. The non-specific probe binding in this ISC model is estimated by the negative WTA probes that can be used to measure such background binding. And finally, we were greatly influenced in this model by some recent work in deep generative modeling or single cell data, as shown in this citation. So I'll jump right into the results. First, what I'm showing you is a good gene that contains very little background that is effectively verified by our ISC model. So what you're looking on the left is the expression profile of both corrected and uncorrected counts. So in blue, you can see that this gene called STMN2 is highly enriched in the cortical plate. STMN2 is a marker of neurons, and we do indeed expect to see high expression of this gene at this time point. Well, I hope you can appreciate that with the ISC correction, which is shown in red. We are effectively sparing this gene from any kind of background reduction, and we're seeing the same pattern. Conversely, what you're looking at here is now a bad gene with a lot of high backgrounds. This gene, DLX1, is actually a marker, a known marker of interneurons. And when we look into human fetal brain, we expect to see the expression of this gene both in sort of the poles of the germinal center as well as the cortical plate. If you look at the raw counts here in blue, you see that in addition to the dashed areas, you see lots of background binding in the middle. And what you have at ISC is the correction of this non-specific background binding in the middle. So in red, you're still maintained the enriched expression of this gene in the germinal centers as well as the cortical plate, but you're not suffering from the negative background binding in the middle. And we indeed validate this with RNA-scope, which is shown on the right. So what does this background correction actually do? So first, it significantly increases the number of genes that you detect from WTA, and we would expect from CTA analysis. But second, it also significantly improves the number of genes that you detect from differential gene expression analyses. So on the right, we are actually showing you DSeq2 analysis of two different kinds of AOIs corresponding to two different cell types of the human fetal brain that we profiled in this study. And when you compare these two different cell types from WTA data, you get a relatively small number of genes, about 200 genes that are different. If you were to use the background corrected counts from ISC, you see that a much higher number of genes are detected that differentially expressed, matching actually what we know about the biology of these two cell types and greatly improving the quality of the data that we have. The second tool that I'll talk to you about is an extension of our efforts to integrate single cell and spatial transcriptomic data. To do this across multiple different kinds of technologies, we've been working in the lab collaborating with Oli Stegel at EMBL to generate a new probabilistic model that we call cell-to-location. And what cell-to-location does is that it can take a cell type reference based on dissociative single-cell RNA-seq, which in this case was a published data set of the human fetal brain, and it can decompose, or if you prefer that word, deconvolve bulk spatial transcriptomic data like nanostring, GeoMx, WTA to give you the abundance of cell types within the GeoMx data. And when we do this, for the human fetal brain, we see really nice, basically spatial identification of cell type locations. Shown here is the proportion of mRNAs from the WTA data that our cell-to-location algorithm attributes to deep layer neurons. And if you look at the spatial pattern, as shown on the right, you see that indeed these deep layer neurons map to the cortical plate where they're supposed to be. And this is work that we are quite excited to share with the community soon, developed by a great graduate student with Vitaly Shevnikov. So what we did is that we took cell-to-location and we actually, with great contributions from Alexander, who is joining me today, we adapted it to account for technical differences in between the WTA data and single cell data, and we used it to basically map single cell type references into WTA. And I think the results were actually quite spectacular. What I'm showing you here today is the spatial locations of the 18 major cell types that is identified in the single cell data set. And the only little bit of biology I'll tell you is that the spatial patterns of these cell types works really great. If you were to look at the germinal centers, we indeed find that different types of neural stem cells are correctly mapped to the different parts of the germinal centers that they're supposed to. And if you were to look into the spatial distribution of neuronal differentiation, we also see a great pattern that validates what we're supposed to see. In the blue box, you can see that the newborn neurons, abbreviated as XN, are located in the germinal centers. The migrating neurons, abbreviated as XM, are sort of transiting on their way to the cortical plate. And then the differentiating cortical neurons are much heavily enriched in the cortical plate, XMU and XDP1. And on top of this, what we can see is that we could take each of these cell types, we could cluster them to many other sort of detailed stratified subtypes, and we also get good information from that. So with that, I'll actually acknowledge the labs, Alexander, who developed both the ISC and the cell-to-location WTA model that I talk about today, Kenny, who did the great work, Robin from NanoString, who was instrumental in helping out with the NanoString experiments, as well as Vitaly and Oli, who worked on cell-to-location. With that, I will pass the baton to Ioannis Vlachos from Beth Israel Medical Center. And thank you.
Thank you, Omer. So hello, everyone from Boston. Before we start, I wanted to mention how excited I am to be part of this e-meeting with so many amazing colleagues sharing their work. I wanted to, first of all, to thank our hosts for bringing everybody together. My group and I are part of the Beth Israel Deaconess Medical Center, Harvard Medical School, and the Broad Institute. And in the hopefully 10 minutes. I will try to share our experience from the integration of spatial transcriptomics with single-cell technologies, some of the challenges, but also the tremendous transformative potential of combining these two modalities. So our group focuses on what we call whole genome and beyond approach. We're trying to extract from a single patient sample the maximum amount of information and integrate it for basic translational and precision medicine applications. However, the advent of two new technologies really reshaped our research program. Of course, single-cell techniques, and this is the traditional single-cell smoothie figure, where enable us to go from bulk RNA-seq smoothies to single fruit experiments. And of course, location. Spatial transcriptomics enable us to put, expression on the map, understand how localization, tissue architecture, and morphology can affect cellular transcriptional programs. And of course, the important part is that these two technologies are exceptionally complementary. From single cell, we get the missing granularity from the bulk techniques, and we can go beyond the literature. We can get our cell clusters and cell types on a hypothesis-free fashion and continue from there. What is also interesting is that even though single cell techniques are rather new, I mean, if we take the history of the world as a reference, there are plenty of data already and methods available for folks to work with. Spatial transcriptomics are the cool new kid on the block and enable us to add architecture and create expression maps, localization, morphology, and understand how structure affects function. So we are utilizing both as a single modality in our work, and we're trying to apply them on a different set of research questions. For the remaining 8 minutes, I will try to provide some high-level strategies and 30,000-foot overview, but also some of those nitty-gritty decisions that can really affect downstream outcomes. I will use as a vehicle for the rest of the presentation a data set on liver we're currently working on. So very briefly, we have human core liver biopsies, and we have the frozen portion where we're subjecting it to SNP-seq, and we have the FFPE, which are subjected to the very cool whole transcriptome atlas on the NanoString Genomics on the DSP platform, where we get 20,000 genes on the ROIs we select. This is an amazing new assay. So in our group, we do not have only data-centric labs as ours, but we have also liver specialists, we have experienced pathologists, and in a collaboration with NanoString scientists, we were able to capture and carve out really informative, really cool ROIs that capture specific morphology features, zonation characteristics, and other liver features we wanted to analyze further. We aim for 16 ROIs per subject, and what is quite interesting, we put 2 subjects per slide for 2 main reasons. One is convenience, of course, and the second is also to be able to assess batch effects and slide-to-slide variation. I will talk a little bit about that because this is something we are asked a lot. For liver specifically, more than any other tissue we worked on, and that could be potentially how the samples were prepared or for reasons unknown to us, we see 2 important effects happening. One is we see clear segregation from slide to slide. So this is a plot showing 4 patients, 2 on one slide and 2 on the other, and they clearly segregate based on their gene expression per slide. But even if we zoom in to just one slide, we see them segregating again in a smaller effect size per patient. Of course, this can be patient-to-patient variation and could be biological, but its effect size is larger than the morphology and zonation we would like, for instance, to examine. So Jared, a brilliant computational biologist from our group, of course, did the first obvious thing. He took, so here we plot only zone 1, 2, 3, with brain orange and purple, and you see the segregation. And he modeled out, slide, and he modeled out the patient effect. And you see, for instance, for the patient, you get a more homogeneous representation, but however, you lose the signal of zonation. So he devised a method where you rescue the biological signal from known and unknown biases, and you continue working with the residuals of that model. So on the bottom, this is not new in computational biology. This has been established by Jeff Leake's group for many years now. And in this adaptation of this approach, we see here the outcome. So these are the BSSR residuals. We're not working on expression space anymore. This is the residual space. We can see that not only similar colored ROIs huddled together, but we keep this natural progression from green, orange, and to purple, which is zone 1, 2, and 3. So this is definitely very exciting. And we wanted to check, how does this affect the biological signal? So we see here 2 heat maps. These are genes in the literature that are relevant to zonation in the liver. And we see, for instance, that in the BSSR, these are the residuals. These are the normalized counts we're using as an input, upper third quartile normalized counts from a lot of help from the nanostring specialists. So we can see the progression from central hepatocytes, zone 1 to 3. These are markers for portal hepatocytes. Again, zone 1 to 3, very gradual decrease. Periportal pericentral endothelial cells 1 to 3, and periportal. If you go to the original, you see, for instance, a mixed signal, and that makes us, first of all, very happy that we are able to salvage this very refined biological signal from, for instance, how the slides were prepared. And we use this residual space to all our downstream analysis. Now this is where the fun part starts, right? We have single cell experiments. And from the single cell, we don't need to use the markers that I just showed that are from the literature. We can find the actual clusters in the pathology that we're studying. We can find subclusters. We can find markers, annotate them, do cellular trajectories. And of course, we can do also cell signaling networks to understand which cells are talking to each other. But having spatial data really can transform what we can do, because from the cell types that we have identified in the markers, we can deconvolve and register them on the ROIs. And that can enable us not only to compare the different zones and different ROIs using gene expression, but also cellular profiles, and can inform. That's why the arrows have 2 edges, and that can also inform the single cell. We can compare zones in the spatial and check which cells are responsible in the single cell. We can, for instance, answer the fundamental question. I have this very interesting macrophage population in my single cell. Are those macrophages in a specific region all together? Are they scattered across the tissue, or are they in specific subregions? And if yes, what are those regions and what other cells are there? And what is the most transformative aspect, at least in my opinion, is that we did before, and as everybody does, right? You have your single cell and you do your signaling network, but you do not know whether those cells are actually adjacent to each other. You can create spatially aware signaling where you can keep the populations. You get your ligands and receptors from the single cell, and you create your network within the ROI. So another thing that we tend to forget, and I wanted to share this with you, is that we focus a lot on the expression space. Everybody does, but we forget that these are images and very informative, where we can, do image analysis, count the cells, apply AI, connect gene expression with cellular morphology and cell counts. And we can also do, image analysis techniques. So let me give you a simple example. So if we see the zonation in liver, it creates two-dimensional vectors that have, different orientation and different lengths. But because we also get, the coordinate matrices for the ROIs in the tissue, we can apply scale and rotation invariant modeling. And that way, we can, for instance, model gene expression as, for instance, distances from specific landmarks, regardless of scale and orientation. There are a lot of things to do on the image plus expression space. I started with this slide and I wanted to close because having both for us has been a completely game changer. We get our cell types and our subtypes from the single cell. We get our very specific for our pathology markers. We have seen a very big difference when using, for instance, atlases from healthy tissues versus atlases we create for our specific pathology and applying them on the spatial. We get our signaling relations and our deconvolution reference. And then on the spatial, we get, the architecture, the map. We can bound cell populations in space, and we can do spatially aware signaling networks. So closing, I wanted to, first of all, to mention that this project that I briefly discussed is part of an extensive effort in the IMC. This is a group effort among 8 PIs. I wanted to shout out to my colleagues, John G., Stefan, Wynn, Yuri, Linus, Gordon, and Jonathan, who are part of this amazing initiative, and of course our lab members who are helping with the analysis. Jared is spearheading this effort, but definitely nothing would have been made possible, if we didn't have NanoString support to bring us close to this technology, provide us access to experienced engineers and scientists, and help us and guide us through this process to really maximize the outcomes and the utility and the potential of our research. So please reach out to us, find us online, find us on Twitter. We have many fantastic things cooking in this space, and I'll be more than happy to take any questions. Thank you.
Thank you, Ioannis, and thank you to all our fantastic speakers that we've had today. I wanted to open up and kick off the panel discussion. We'll start with you, Ioannis, since you just closed us out. And I wanted to talk to you a little bit about the vision that you see for where Beth Israel is going to take these platforms in the near term, and maybe what the long-term vision for integrating these different types of platforms would look like to you.
So, yeah, first of all, that is a big question, to be honest. So this is something we discuss on 2-3 different levels. We discuss it, within the lab and our colleagues on the hands-on, boots on the ground. But we also discuss it with our clinicians, because it is a research slash academic slash clinical environment, and we also discuss it with our leadership. So my understanding is that definitely for research, this is transformative. And I see a lot of colleagues, they see our first results and they want to test the water. They want, since we already have samples in the freezer and we already have FFP blocks, how can we revolutionize what we see and increase granularity? But also, we are discussing with, clinical trials, and we're discussing with, how can we bring this to inform clinical decision-making? How can we inform the drug pipeline? I see within the next, let's say, 1 or 2 years, definitely research will incorporate more and more, but within the next 5, I see more trials. And I think, and I hope that BI will be spearheading this effort.
Absolutely. I feel like that's one of the reasons why we strive to have such a strong relationship with your team, because we see you guys actively working and developing towards that. I wanted to talk a little bit more granularly about the near-term needs that your research team has. You've talked a little bit about some of the methods development and some of the thoughts that you're putting in terms of using the spatial data to inform some of the metrics that you're getting out of single cell data. And do you have any thoughts about where that's going in the short term and what methods need to be developed to enable that more robustly?
Yeah, so for us, that I do not know for sure, because for us this is, and for everybody working with these new technologies, especially the whole transcriptome atlas is new for everybody. Our first approach, and I think this is where things are going, is exactly to understand the commonalities and the differences between existing modalities. It looks like RNA-seq, but it isn't. How does that affect our model? This is very, very simple. It might sound very simple, but if we want to make elegant modeling, understanding exactly the distributions, the limitations, and how to incorporate simple things, the negatives that you have, so that is one thing. Maximizing the information we can get from the raw data is one. And the second is, can we do this seamlessly? Can we map, for instance, do transfer learning, and can we map our single cell with our spatial data and integrate them very easily? Can we anchor the cells that we find? What is the limitation on the granularity? For instance, for liver, we have 52 clusters. Should we try to automatically map them all? Is there a level of granularity? And then imputation methods, can we use the spatial impute missing genes in our... these are just low-level practical things. And these are the very short term. I think we can dream bigger as soon as we have those sorted out.
Absolutely. I have one more question for you quickly. In order to enable those types of development efforts around understanding the data types and the data methodologies, what critical holes in terms of access to data do you see? And what you'd mentioned that there was, some potential roles for developing atlasing efforts. Do you see that playing a large role in the short-term build that you need to be able to then move towards some of those longer-term goals that you have?
Yeah, so I'll try to very quickly, but this actually has a lot of branches. We see this, for instance, in BI, and I'm pretty sure that BI will be spearheading this because we have a share and open communication channel across the institute, and that bringing more people on board definitely is a benefit, especially for a new modality. If we extrapolate this to the community, then yes, and as I mentioned, it's not just the counts, right? So for one range of applications I would love to see would be to maximize the utility of the image data at the same time, use AI, but you need the images for that. You need the raw data for that. You need access to everything, which will usually not be part of a supplement. So having access and being able to compare everything across groups, I think it will be transformative for the community. And that's how single cell actually moved so quickly. I mentioned in the presentation that despite the very few years, we have a ton of tools. Human Cell Atlas has been instrumental for that. If spatial moves towards the same direction, I think it will get the mass and scale very quickly as well.
Thank you so much. And to kick off the panel discussion, I'd like to return to Dr. Chris Mason. Dr. Mason, since you've been working with the COVID Immune Response Atlas, I wanted to ask you a little bit about how you're thinking about, with all the great data that you collected from other sources, how you're planning to integrate that over time and what lessons each of the data is telling you in conjunction with each other.
Yeah, great question. So I think, the integration between bulk data, single cell, and spatial transcriptomic data has been powerful to see, but just in a quantification level is one thing. We confirm what we see for the presence of the virus. We can also confirm the host response, what's happening to the cell. So they at the very least serve as a validation. But I also think if we can get, what we started doing is taking even the spatial data when we do need to capture, what you get when you actually cleave is you will get information also of what's present, in terms of some of the alleles that might be there. So we started to look at that as well, even things like allele-specific expression, and also just looking at the variants in the virus, started to do that as well. So I think the integration's just started across those modalities and has been consistently hitting interferon networks and also immune system stress, which, unfortunately can lead to some of the patients that we did the autopsies on. But at least we can learn about what the targets are in cells present for other drugs that we could use to help them.
Fantastic. And on a similar note, Dr. Curtis, I wanted to think with you or hear from you about how you're planning to take the genomics data that you've got right now and think about, from a translational perspective, what new data types and data sets you'd like to be collecting to augment the data that you've got right now, as well as bridging out and building out larger cohorts in future studies.
Sure. Yeah. So I didn't get into the details given the short timeline, but we've been really encouraged by how well we've been able to resolve differences at a proteomic and signaling level between responders and non-responders, and in fact have shown that we can reliably predict PATCR and build a classifier to do that. Which validates in an independent cohort. So we're pretty encouraged about the spatial proteomic readouts. And as I alluded to, there's signal there that we simply don't see in the bulk transcriptome. So I think one of the next steps that we're really excited to take is to leverage the cancer transcriptome panel, which has matured considerably and is now, I think, would really allow us to go back in and ask, whether the differences that we observe are attributable to the spatial context or if it is the more proximal readout, perhaps, of looking at the protein level. So that's something that we're really excited about, and also seeking to incorporate some single cell transcriptomic data again to map in as much as possible in an unbiased way what these differences look like across therapy.
And in your institution's approach, are there any special considerations that you're thinking about while you're planning those studies?
Sure. So we have a number of studies planned, both in clinical cohorts, often longitudinal cohorts, but also, retrospective collections. And I think one of the key elements that we're always considering and paying attention to is how we go about sampling the tissue, setting it up in a way that we can really do high throughput studies. So one aspect of that that's been essential is having a pathologist involved really early on. In many instances, as we go forward with larger cohorts, we're also embedding these tissues on tissue microarrays so that we can ensure the sort of batch correction across multiple samples from the same individual, but also just throughput wise. And I think one of the important findings that we've had is that even with relatively small core biopsies or core punches, we're capturing enough of the tumor microenvironment to really delineate some of the fundamental differences there. And I think that's something that, up until now wasn't totally understood how deeply we need to sample. So we paid a lot of attention to that. And we're also, in collaboration with NanoString, thinking carefully about the phenotypic selection strategies and how we can really harness the DSP technology to perhaps delineate particular lineages before going deeper with the cancer transcriptome panel and the whole transcriptome panels.
Thank you. No, that's great perspective that you've offered there. You mentioned the role of pathologists and what they bring to the table while you're doing and setting up the experimental design. I know that Dr. Mason has been spending a lot of time and worked really closely with a group of pathologists at Cornell Wheel during the experimental design for the genomics experiment. Would you mind talking a little bit about that, Dr. Mason, in terms of what role they played and how it informed the study design that you had?
Yeah, absolutely. So I think this was a real team effort, not just my lab, but also, this is with Rob Schwartz and also Alan Borsuk, who's a pathologist here. And it's really, it was really strange in the early days of what could we do for the autopsies. We didn't know, for example, using the bone saw, on the skull was not allowed because it aerosolized too many components of the body. So autopsies had to be done very carefully. It was a lot that was unknown in April and May as to what we could even expect for potential pathogenicity of the virus post-mortem. But Dr. Borzik, Dr. Schwartz, like really it was a team effort of the three of us, and also our labs all working together, and also getting samples and bulk RNA sequencing and single cell sequencing to match this. So it was a really team effort, and so I really represent all of our work here. And it was a great example of staying in touch with people who do the autopsies and pathology work at your hospital, because you never know when you may need to make a phone call to them and say, hey, what samples do we have? We should really look at this question from a spatial perspective to complement the other RNA data we were getting. And it was a great team effort.
It's fantastic to hear. And I know that some of the other members on the call as well have been interacting pretty closely with their pathology. So one of the things that stuck out to me, Omer and Alexander, with the WTA data that you were working through, was that you've been really thinking deeply about the shape of the data and the type of the data that you're getting to work with. I wanted to ask you if you could provide a little bit of context for some of the analytical considerations that went into building your model and some of the concerns that people might have when they're approaching this new data type.
So, sort of a high-level answer that I can give is that there's actually plenty of similarities that you can find in how people approach single-cell data and how you can approach WTA data. So the analytical frameworks that we've been using with the deep generative models, assume that a negative binomial distribution was a core part of nanostring WTA data. But when you think of a multi-spatial scale data set like nanostring, where you can specifically profile select cell types in certain cases, as few as 5 cells or single cells, or you can profile much bigger bulk ROIs where you have thousands of cells, much higher numbers of transcripts, much higher number of genes. You're talking about really like a vast distribution. And what we've found is that flexible approaches might be required. Alexander has had great success using actually gamma distribution when he was looking at really large ROIs and really highly expressed genes. So I think with WTA and possibly with CTA, the analysis approaches have to account the vast heterogeneity of different types of spatial ROIs, AIs that you can do. When it comes to integration with the single cell data, what we've noticed with WTA and other methods like Visium, slide sequencing, et cetera, is that, and what we and others have noticed, is that the platform-specific effects are a huge component and obstacle to proper integration of the data sets. So, if you basically took, let's just say that you processed adjacent tissue sections with technique 1 and technique 2, you are going to end up recording different quantities of select genes. And that will stem to myriad technical reasons of how the different assays work. And I think any kind of integration tool that aims to combine those data sets together has to account and model those technical differences. And doing sort of like bulk correlations between different technologies like single cell and WTA is always going to be error prone at some point.
Thanks for that particular background, and I completely appreciate that that's a really critical part of validating that the data and the model that you're setting up is valid and is going to give you the context that you're interested in. So I want to think from a broader perspective about someone who's coming into the field with no real background in spatial biology. And I wanted to talk through this with each of you about what your perspectives are that they should know before they start doing data analysis for spatial omics data, and how they should think about this as being complementary or a novel data set type that brings different perspectives to the field. Chris, can we start with you on that one?
Sure. So I think there are some tools already existing in open source tools. NanoString has tools on the computational side that are the first thing to get familiar with. The other thing is, it's like anything in biology, it's very exciting to see such high resolution data, to really look at, unparalleled, multiplex tracing of, thousands of genes across tissues. But at the end of the day, the bedrock of good science is replicates, right? So you can't just do 2 or 3 slides and be like, okay, good, we're done. Even though it's the data is deep and rich and complex, and it's a little bit expensive, you need to plan for a proper experimental design, which means multiple samples with validation and replicates. So I think those two, the computational and experimental design features, are important, but they are things that have been established for other fields for a long time. So I'd say get familiar with those first, and then also some of the protocols. And also there's some customization you can do with the probe. So I think if you have something unique you want to do, at least in our experience, been very collaborative with NanoString. So, I'd say for people that have a weird idea or hypothesis, something they want to look for in a cell, you should ask for it, because it's probably something that's doable. Within reason, I guess I'd say. I can't speak on behalf of NanoString, but within reason, yeah.
We try to be good partners on that.
Yeah.
Christina, what are your thoughts about someone new coming into the field and what they should expect from spatial omics data?
Sure. So I echo a lot of Chris's sentiments, and I think that study design is crucial. And thinking through, it's great to have these new tools, but what is the hypothesis that you're after? What's the question, that you're seeking to address, and designing the study accordingly is obviously key. I think from a spatial perspective, it's obviously important to do the initial QC up front on any data type that you have, to the extent that you have orthogonal measurements that you can corroborate findings against. I think that's quite important too, but obviously not always feasible. And I guess the other point that I would just call out is I think that, now we're beginning to have the capacity to look both spatially and temporally. And I guess I would just hearken to the fact that I think that the longitudinal comparisons also call out some additional considerations, and perhaps though, to say that with spatially resolved approaches, we're mitigating some of the challenges that we see when we're trying to compare bulk populations, admixed populations through time, when there's changing numbers of cell types and so forth that are hard to sort of pin down. So I think that there's a real opportunity to harness the spatial approaches as we think about time series data, as it were.
Fantastic. No, I completely agree. And it'll be really interesting to see these data sets evolve over time as more of them come online and become available to people. Omer and Alexander, did you have any perspectives that you wanted to share on how your experience with NanoString WTA data has evolved over time, and what lessons might be appropriate for new people approaching the platform?
I think Alexander could take this one as the person who did work with WTA data directly. A question. As somebody who was very experienced in single-cell transcriptomic data analysis, how did you find that your skills translate to WTA data? And do you think, being well-versed in single-cell work makes you a better WTA analyst, or is it a completely different ballgame?
Oh, I see. It was in many ways similar. The whole structure of the data, it has unique molecular identifiers, so that's very useful. That's similar to single-cell RNA-seq data. And then, the version we used had the whole transcriptome, sort of information from the whole transcriptome available. So that's, again, a little bit similar. So in terms of the models we were developing for this data, actually, it was mainly the background that was a little bit different. So in single-cell RNA-seq data, we have this thing called soup, because cells can break up and we can get a bit of RNA from other cells. This is one type of background in single-cell RNA-seq data. And then in WTA data, we have the background binding, which we need a slightly different model for than the single-cell RNA-seq data. And then the sensitivity was actually quite comparable, so that was great. So we find a similar number of genes detected, sort of in compared to most single-cell RNA-seq data sets. Yeah.
Fantastic. And let me give a few more seconds there. Since we didn't get a chance to talk to you, Alexander, earlier about your model when we cut off, I'm going to phrase a question towards you, and then maybe you can jump back in on that. So when you were working on building your model for analyzing the genomics WTA data, what were some of the considerations and background that you took from other approaches that informed the model that you developed, that you were working with the WTA data?
Yeah, there was already quite a bit available. People have thought for a while about things like sampling noise that we get from technologies such as RNA sequencing or so, because we never actually catch all the RNA, whatever assay we're using. And this process itself creates a little bit of noise because we only capture maybe 1%, 5% of the RNA in the cell. And people have also thought about generally, how can we summarize the expression of a cell biologically using deep neural networks or using something simple such as principal component analysis, factor analysis. So we could reuse these parts for WTA. Then WTA has negative probes available. This gave us some information about the background binding, which we need to account for in WTA. And then WTA, yeah, it doesn't have quite as many samples usually. I mean, the typical experiments as single-cell RNA sequencing, because in single-cell RNA sequencing, we can sometimes have millions of cells even, sometimes a few thousands. So in a typical WTA experiment, maybe we have information from 50 or 100 samples, maybe soon more. And then you sort of need to adjust the models a little bit so you don't overfit the data, and you need to make slightly less complex models. Mm-hmm.
Great. Thanks for that perspective. That's really really important to keep in mind, as this data has so much ability to fit the type of model and type of experiment that you want to do. It has that flexibility in the ROI selection. That does bring up another point that I wanted to poll the room a little bit about, which was the role in which developing broader access data sets might help inform the community, and what role that maybe some of you work on in terms of cross-institutional consortia might play in developing new resources for the community. I know, for example, Christina, you're part of HTAN, and I was interested in your perspective on how the community might collaborate in the future on these types of projects.
Sure. So within HTAN, there's which is the Human Tumor Atlas Network, there's been fairly extensive efforts to try and generate orthogonal spatial and single cell resolution data sets on the same material as a means to inform the development of analytic tools. And in some ways that's still plugging along. And I think that as we've heard from many of the speakers, right, a big push going forward is really going to be on the analytics and how we interpret and leverage the most information from these rich biological data sets. I certainly think that there's value in establishing standards and best practices, both for approach, but also for the downstream analytics. And that, of course, takes time. You know, we need to spend time with the data to understand what's optimal. And this is still a very nascent field. I also believe, though, that as you point out, right, having sufficiently representative cohorts that have been profiled, right, that allow us to understand the composition, topography of different cell populations could be really valuable. Clearly, there's going to be context dependencies, and I think in cancer, that's something we wrestle with a lot. There's the tissue dependencies, there's the temporal and the state of disease. But I do think having reference data sets could help on many fronts in terms of moving the field forward.
Chris or Omer, anything you'd like to add to that?
No, I think some of the, I guess I'd say some of the things I'm also looking for, some of the better measures of heterogeneity that we're seeing themselves can be a metric. We've seen some of this from the bulk field, is that heterogeneity itself, epigenetically especially, can indicate, have a prognostic value and indicate risk. And so I think we'll start to see this probably also come out of the spatial data as more data comes out.
Great. Thank you all so much. I think we're getting pretty close to time here for you. So I think we're going to wrap up our panel discussion. I want to thank all of our speakers for the fantastic conversation that we've had today, and I hope that this has been a thought-provoking exploration of the analytical needs and the future for spatial genomics to integrate with the analytical toolkits that people have available for their next waves of scientific exploration. In addition to the great work described by the panelists here today, I'd like to point out that NanoString is excited to announce the launch of the Genomics Translational Leadership Network, the GTLN, which is aimed at, again, helping develop some of these shared resources, tools, and facilitating conversations across large institutions, and has been working with breast cancer researchers in one case, as well as COVID researchers in another case. We are now starting to take applications for new members to build future consortia in oncology, immunology, and neuroscience, spanning the realms of discovery science as well as translational and clinical applications. Applications can be found on our website, nanostring.com, and stay tuned for more GTLN updates, or please contact geomics@nanostring.com to learn more about Geomics DSP or GTLN. Thank you all again for the fantastic conversation today and for your participation.